Topic of presentation is lipid metabolism. Fatty acids are the derivatives of lipids. Fatty acids contain a long chain hydrocarbons, and on its terminal end a carboxyl group is attached. The hydrocarbons may be saturated with no double bond or may be saturated containing double bond. Fatty acids can be obtained from diet, adipolysis or de novo synthesis. Oxidation of fatty acids. Fatty acids are oxidized by most of the tissue in the body. However, brain, RBCs and adrenal medulla cannot utilize fatty acids for energy requirements. Digestion of fatty acids. Minor digestion of triacylglycerol occur in mouth by an enzyme called lingual lipase, and major digestion of all lipids occur in the lumen of duodenum and duodenum by an enzyme called pancreatic lipases. Mobilization and stress. When body needs energy, fatty acids are released from adipose cells and mobilized for use. The process begins when the level of glucagon and adrenaline in the blood increases and these hormones bind to specific receptors of the surface. Here you can see this hormone is binding with a specific receptor on the surface of adipose. This is the adipose cells, and here it binds. Transport of fatty acids. Free fatty acids are also called unesterified or non-esterified fatty acids, and their states are called unesterified states. In plasma, longer chain free fatty acids are combined with albumin, and in the cell they are attached to a fatty acid binding protein. Shorter chain fatty acids are more water soluble and exist as the unionized acid or fatty acid anion. By all these means, fatty acids are made accessible as a fuel in other tissues. Triacylglycerol, an ester of glycerol with 3 fatty acids. This is called triacylglycerol triglyceride. This is a simple category of a lipid. Biosynthesis of triacylglycerol. There are 3 steps involved in biosynthesis. First is synthesis of glycerol phosphate. Second one is the conversion of free fatty acids to its activated form. Third one is synthesis of molecule of TAG from glycerol phosphate and fatty acyl-CoA. First is synthesis of glycerol phosphate. It occurs in liver and adipose tissue. First of all, glucose in both converted into dihydroxyacetone phosphate by a process called glycolysis. In the next, enzyme glycerol P dehydrogenase acted on it and converted it into glycerol phosphate. Here, the synthesis of glycerol phosphate occur. In the next step, conversion of free fatty acids from its activated form. Here you can see in the intestinal mucosal cell, all these fatty acids, fat-soluble vitamin, 2-monoacylglycerol, long-chain fatty acid or cholesterol, all these are in free fatty acids form, in their free form. And by different means of enzyme, here you can see, they are converted into their activated form in the chylomicron. In the chylomicron, they are in their activated form, and from there they go to lymphatic system and do their work. The synthesis of molecule of TAG from glycerol phosphate and fatty acyl-CoA, and the glycerol phosphate is converted into triacylglycerol. First of all, glycerol phosphate is converted into lysophosphatidic acid. From the lysophosphatidic acid, it is converted into phosphatidic acid. Phosphatidic acid is converted into diacylglycerol, and from the diacylglycerol, it is converted into triacylglycerol. Here, different enzyme acyltransferase, phosphatase or acyltransferase is present, which converted it into and in different processes. Triacylglycerol cycle. Triacylglycerol cycle occur in adipose tissue, blood and liver. In adipose tissue, triacylglycerol is present, which is converted into fatty acid, and here a glycerol molecule is released from it. And when the fatty acid is converted again into triacylglycerol, a glycerol 3-phosphate is added into it. In the liver, triacylglycerol is also present. It is converted into fatty acid in the adipose tissue, and in the blood they release glycerol. Fatty acid is again converted into triacylglycerol in the liver, and their glycerol 3-phosphate is added into it. When the fatty acid goes from blood to the liver, they use as a fuel. Here As we discussed earlier, fatty acids are used as a fuel in different tissues. Activation of fatty acid. The first step in the activation of fatty acid is the activation of free fatty acids. It must to activate fatty acid before being catabolized. Here the two steps are involved, first and second. Here the fatty acid is converted into acyl-CoA by the mean or by the presence of an enzyme thiokinase and in the presence of an enzyme ATP. Three different thiokinases have been identified to long chain, medium chain and short chain fatty acids. Transport of acyl-CoA into mitochondria. The inner mitochondrial membrane is impermeable to fatty acid. A special carnitine carrier system operates the transport of activated fatty acid from cytosol to mitochondria. The carnitine is converted into acylcarnitine. This step is present in the cytosol. And here is the mitochondrial matrix. Here they tell us matrix. From the acylcarnitine, a carnitine carrier system help it to move from the cytosol to the mitochondrial matrix. In the acylcarnitine in the mitochondrial matrix, in the presence of carnitine, acyltransferase 2 enzyme is again converted into carnitine, and from here it goes to or refuse into the cytosol again. This is the translocation process. Beta oxidation. Most of the fatty acids in the body are oxidized by beta oxidations. It is defined as the oxidation of fatty acids in beta carbon. The result in the sequentially removal of 2 carbon unit acetyl-CoA. Bioenergetic of beta oxidation. In each cycle of beta oxidation liberates 2 carbon unit acetyl-CoA. It starts off carboxylic ends. It occurs in a sequence of 4 reactions: oxidation, hydration and cleavage. The acetyl steps of beta oxidation: first, second, third and fourth. In the first step is oxidation, second step is hydration, third step is cleavage and third step is oxidation again, and fourth step is cleavage or thiolysis. In the first step, acyl-CoA is converted into trans-2-enoyl-CoA. In the next step, hydration, the trans-2-enoyl-CoA is converted again into 3-hydroxyl-CoA. 3-hydroxyl-CoA. 3-hydroxyacyl-CoA is again converted into beta-ketoacyl-CoA. In the cleavage, it is converted into acyl-CoA. The acyl-CoA formed in the cleavage re-enters the oxidation pathway at reaction 2. Since acetyl-CoA can be oxidized to CO2 and water via the citric acid cycle. Here is the overall reaction of beta oxidation. You can see here. This is the overall reaction. The next is omega oxidation. It involves hydroxylation and occur in the endoplasmic reticulum. Hydroxylation takes place in the methyl carbon. It uses the mixed functional oxidase and reaction requiring cytochrome P450, O2 and NADPH. Hydroxy fatty acids can be further oxidized to a dicarboxylic acid via sequential reaction of alcohol dehydrogenase and aldehyde dehydrogenase. The process occurs primarily with medium chain fatty acids. Omega oxidation pathway. There is another pathway in some species including vertebrates that involve oxidation of omega carbon. The enzymes unique to omega oxidation are located in the ER of liver and kidney. The preferred substrate are fatty acids of C10 to 12 carbon. Here they are the 10 to 12 carbon which are involved. You can see there. In the first step, it introduces a hydroxyl group onto omega carbon by mixed function oxidase and involved in cytochrome P450 and NADPH produce a carboxyl group at each end. You can see a carboxyl group is produces. In each pass through the beta oxidation pathway, the double-ended fatty acid yield dicarboxylic acid such as succinate or adipate is formed. Fatty acid yield dicarboxylic acid such as succinate which can enter citric acid cycle. This succinate is after this added into citric acid cycle. In the next, biosynthesis of saturated fatty acid. Fatty acid synthesis occur within the cytoplasm of animals. Fatty acid synthesized when the diet is low in the fat or high in the carbohydrates and proteins. acetyl-CoA, which is present in the mitochondrial matrix, cannot go or refuse from the mitochondrial matrix. It is impermeable for this, so it is converted into citrate, and the citrate goes to cytosol and it is converted into oxaloacetate, then malate, then pyruvate, and this pyruvate is again from the cytosol go to the mitochondrial matrix and converted into acetyl-CoA and oxaloacetate. So, this is the biosynthesis of saturated fatty acids. In the next one step, palmitic acid. A palmitic acid is 16 carbon chain acid or fatty acid. Here is its overall reaction. You can see 8 acetyl-CoA, 7 ATP, 14 NADPH and form a palmitate and other different bioproducts. Raw material for palmitic acid, or it is also called de novo synthesis. In this, trans-enol ACP is converted into acyl ACP, then acyl S enzyme, then acyl ACP, and in the last it is converted into palmitate. Different steps are involved in it: enol reductase, transfer of C chain to cis, SH, 6 more cycles of reaction 2 to 6 or thioesterase. FAS. Fatty acid synthesis begins when the carboxylation of acetyl-CoA is converted into malonyl-CoA. From the acetyl-CoA, malonyl-CoA is formed in the FAS. It is a complex dimer comprising 2 identical monomers containing all 7 enzyme activities of fatty acid synthesis on one polypeptide chain. The use of one multi-enzyme functional unit has the advantage of achieving the effect of compartmentalization of the process in the cell without direct permeability barrier. FAS complex. It is a multi-enzyme complex dimer composed of 2 identical subunits. Its weight is 2,40,000. Each subunit contains the activities of 7 enzymes of FAS and an ACP with 4 phosphopentadiene SH group, and the 2 subunits lie in antiparallel. Here you can see FAS synthesis. Fatty acid synthesis is formed by here, and multi-enzyme means different enzymes are involved in whole this reaction, so it is called multi-enzyme complex. Comparison of beta oxidation and FAS. First of all, its location. Beta oxidation pathway is present in mitochondria and fatty acid synthesis occur in cytoplasm. Acyl carrier in beta oxidation is coenzyme A, and in FAS is 4-phosphopentadiene or cysteine. Electron acceptor and donor in beta oxidation is FAD and NAD, or in FAS it is NADPH. OH intermediate in beta oxidation is L, and in the FAS it is D. 2 carbon product or donor in the beta oxidation is acetyl-CoA, and in the FAS is acetyl-CoA or malonyl-CoA. Ketone bodies are metabolic products that are used or produced in the excess during excessive breakdown of fatty acid. And acetone, acetoacetate or 3-hydroxybutyric acid. These are collectively called ketone bodies. They are water soluble and energy yielding. Biosynthesis of and ketone bodies. First of all, acyl-CoA is converted into acetyl-CoA to acetyl-CoA. Acetoacetyl-CoA and acetyl-CoA. From the acetyl-CoA it is converted. And in the next 3-hydroxy-3-methyl-glutaralcyl-CoA is formed. And from there acetoacetate, and in the next acetone and 3-hydroxybutyrate is formed. And collectively these are called ketone bodies, and by this a ketone body is formed. Ketone bodies utilization and role in tissue. Ketone bodies are one of the fat burning in the body. When there is not enough insulin, your body is unable to use sugar for energy, and your body breaks down its own food and fat and protein. When fat is used, ketone bodies and acid and appear in your urine and blood. And adipose tissue, triacylglyceride is converted into fatty acid and go to peripheral tissue and in the liver. In the liver it is converted into acetyl-CoA, then acetoacetate, then beta-hydroxybutyrate. And beta-acetoacetate and beta-hydroxybutyrate is go to peripheral tissue. Adipose cell tissue with the fatty acid which goes from adipose tissue to peripheral tissue is converted into acetyl-CoA. Or acetoacetyl or acetoacetate or beta-hydroxybutyrate is converted into acetyl-CoA, and in the last it is converted into carbon dioxide and water. Cholesterol metabolism. Next one is cholesterol metabolism. It is present in animals, sterols, it is amphibethic in nature, or organs which are involved in the synthesis of cholesterol is cytosol, micromers of adrenal cortex, liver, intestine, testes, ovaries, skin, and adrenal cortex. Biosynthesis of cholesterol. Biosynthesis takes place in the endoplasmic reticulum of hepatic cells and begin with the acetyl-CoA, which is mainly derived from an oxidation reaction in the mitochondria. In the acetyl-CoA, this comes from mitochondria, and all these reaction is done in the ER or endoplasmic reticulum. Here 3-hydroxy-3-methyl-butyl-CoA is converted into mevalonic acid, then mevalonate 5-phosphate, then mevalonate 6-pyrophosphate, then isopentyl pyrophosphate, then granyl, then farnesyl, then soqueline, then 2,3-oxydisoqueline, then lanosterals, then cholesterol. It is the whole process or whole function, whole biosynthesis of cholesterol. By this, a cholesterol is formed from acetyl-CoA. Regulation of cholesterol synthesis is hypocholesterolemic drug, fasting, glucagon, glucocorticoids, increased dietary cholesterol. Here is the overall reaction of cholesterol synthesis. Here I will tell you about these. Cholesterol synthesize or regulate these 4 things. Next, steroid hormone. Steroid hormone with 8 to 10 carbon atoms in the side chain of alcohol and alcohol hydroxyl group at C3 are classified as sterols. You can see 8 to 10 carbons or OH or alcohol hydroxyl group at C3 carbons. This is a steroid hormone or steroid structures. Utilization of cholesterol synthesis of steroid hormone. First of all, utilization of cholesterol. Cholesterol is utilized into pregnenolone, then progesterone, then cortisol, then aldosterone, then estradiol. In the progesterone, it is converted into these 3. And the synthesis of steroid hormone, you can see acetyl-CoA is converted into acetoacetyl-CoA. All these reactions we discuss in the biosynthesis of cholesterol. By these, from the acetyl-CoA, a cholesterol is formed, and from the cholesterol, a steroid hormone is formed, or vitamin D or bile acid is formed. Thank you so much.